Hello, and thank you for giving me a chance to present our work today. My name is Dirk Dittmer. I'm a professor in the Department of Microbiology and Immunology at the University of North Carolina at Chapel Hill. And over the last 2 years, we've been involved in sequencing genomic variants of SARS-CoV-2 in the state. We've also been sequencing a lot of other viruses, and today I will summarize the lessons learned from our endeavor. These are some required disclosures. I leave them on for a little while. The research is funded exclusively by the Centers for Disease Control, the state of North Carolina, and the University of North Carolina Cancer Research Fund, as well as the NIH. We're interested in sequencing viruses, both large and small, and I will summarize work that we have published over the last 2 years on SARS-CoV-2, as well as on a DNA virus called Kaposi sarcoma-associated herpes virus, and then new data that we have generated since the beginning of the pandemic. Now, the thing about virus sequencing as opposed to sequencing human cells, for instance, that no two viruses are the same. The example I give here are severe acute respiratory syndrome coronavirus, SARS-CoV-2, which is an RNA virus. It's relatively small, about 30,000 base pairs, and it's present at very high copy number, up to 10 to the 8 copies of the genome per mil in the nasopharynx. Kaposi sarcoma-associated herpes virus, on the other hand, is a DNA virus, and it's a large DNA virus. It's about 5 times to 6 times larger than SARS-CoV-2, and it's present in relatively low copy number in blood. And so these two properties present different challenges. We didn't come out of the blue, but we've been working on the diagnosis of viral viruses since the lab 2010. In 2010, we got into a comprehensive analysis of West Nile virus. And the key insight from these earlier studies is that we were able to generate PCR products that tile the entire viral genome. On the panel A on the left, you will see matched PCR primers that give a product of the same size, around 400 base pairs. And then on the right, you will see the same primers, but now combined differently so that they span the entire viral genome. We use the same approach then for KSHV. Here the problem is that the virus is much larger, and we still want to be able to put all the primers in the same tube, and so the design approach is much more complicated. We were relying on a technology called AmpliSeq that generates overlapping primers. For KSHV, there is a unique central region that we were able to amplify of about 100,000 base pairs. What I would like to consider next are 4 or 5 important aspects of sequencing. I will show you that primer-directed AmpliSeq outperforms randomly primed RNAseq, that the viral load determines coverage, which is kind of intuitive, since the more target you have, the higher coverage you can get, that the amplicon length determines the sensitivity of the assay, that there is a possible primer bias in amplification, in that some primers will perform better than others, that there is a possible primer bias in the sequence alignment. And so one of the advantages of the AmpliSeq enzyme chemistry is that it digests the primers before library build. Otherwise, you would have to use bioinformatics to remove the primers. And I will tell you what is also self-evident, that there are always gaps in any approach trying to tile across the genome, and how do we deal with these gaps. Our sequencing pipeline was designed to generate results within 48 hours. If you push the people and the machines within 24 hours. The startup is very conventional. There is an inactivating step with tween, which eliminates infectivity. There's a step to digest nasopharyngeal secretions if they're there, like the mucins. That step is not needed for blood. There are commercial ways to isolate both DNA and RNA. They're certified for total nucleic acids. The Roche one is automated, and the pipetting is automated on an open source system. The AmpliSeq technology is commercial. We use two sequencers, either the Ion Torrent Genexus or the Ion Torrent S5, and then we use Sanger sequencing to fill in the gaps. Likewise, the bioinformatics tools are pretty much standard, and every complete genome we get is uploaded to GISAID, the international database that collects sequence information on all SARS-CoV-2 variants circulating around the world. And they have requirements in that there should be less than 1,000 undetermined nucleotides. The first point is that targeted sequencing delivers even coverage. And we're comparing in panel A regular hexamer primed RNA sequencing, and in panels B, C, and D AmpliSeq sequencing on consecutive dilutions of input. You can now see two things. One, we can dilute the input up to 40 folds and still get the same amount of coverage. And two, comparing A and B, the coverage is dramatically higher if we use targeted amplification. You can also see, comparing A and B, that the coverage is more even, and that in hexamer and poly-DT primed sequencing, you have good peaks at the beginning, at the end, and then you have dips in the middle. And also, if you look at the blue line, the spikes are much higher. So that is the advantage that AmpliSeq or targeted sequencing provides. And we're using that now even for RNAseq of human genes. The ability to call single nucleotide variants across a wide range of dilutions is crucial, because we never know how much virus is in a given sample, and we don't want to bias our analysis for samples that have had very high viral load. And so here we have two comparisons. One is a wild type sequence that has 8 variants compared to the wild type, and we can dilute that 16,000-fold and recover them all. And in the top, we're using a mutant sequence that has 428 artificial variants, and we can dilute that up to 16,000-fold and still recover most of the variants. It is important, though, that viral load ultimately determines sequence coverage, and the CDC has given a guideline for SARS-CoV-2 that typically CT values, which is a measure of viral load, should be less than 28. These two panels are from a report of 2020. You can see here on the y-axis the log number of mapped reads and the CT value on the x-axis. There are two primers. One is RNAseq, which is not viral, and you can see there's no correlation between mapping to the viral genome and a cellular primer. And on the left N1, you can see a very nice linear correlation. That is, as soon as you hit about 30 for CT, the mapping is linear correlated with viral load. So mapping to a genome is a very expensive way to determine viral load. On the right, you're comparing the number of mapped reads versus the percent coverage. And you notice that you need about 10 to the 5 mapped reads in case of SARS-CoV-2 to get 100% coverage. That is coverage of about 5x. Now, there are a lot of samples that are positive and will give some mapping to the genome, but not complete coverage. And that is still useful. Here we are looking at some very weakly positive or negative samples. The white panels on the top show you samples where fewer than 20 reads mapped to the SARS-CoV-2 genome, and clearly these would be random artifacts of mapping. The bottom panel shows you mappings of 60 to 800 reads. Clearly not enough to call mutants, clearly not enough to sequence the virus, but demonstratively much better than the negative samples. And so this is an independent confirmation that even these weakly positive samples still had virus in there and had complete virus, because you can see the mapping is random across the genome. Positions are on the x-axis. This is from a rather old paper where we asked a very simple question: does the length of the PCR product determine the sensitivity of the PCR? And so you can see the CT values on the vertical axis and the amplicon length on the x-axis. Up to about 500, which is in the middle, the CT values are the same, independent of amplicon length. And then on the right, as you go from 1042 to 2650, you can see an almost linear increase in CT values, which is equivalent to a loss of sensitivity. I also indicated the average primer or amplicon length of the PCR collections that we're using. That is, SARS-CoV-2 is much smaller than the SARS-CoV-2 Arctic primers or the case HV primers. This is nothing new. It's something that we're keenly aware of in viral diagnostic. We know, and I just told you, that random sequence prime gives you nucleotides in the range of 50 to 800 pieces. AmpliSeq with 237 amplicons, the length is 200 nucleotides. Arctic, which is another approach for targeted amplification of SARS-CoV-2, has fewer amplicons and they're longer. And they perform well to a CT of 30, which is about 10,000 copies of the target. Now below that, I have diagnostic PCR primers that are used by the CDC or the WHO. They have sizes ranging from 67 to 113, and they're able to detect targets down to 1,000 copies. So going from 200 to 100 gives you a lot more in sensitivity. Now for comparison, I provide you the sensitivity of the FDA-approved HIV assays. The first one took 5 years to develop and went down to 400 copies. The next iteration, COBAS, took 10 years to develop and went down to 40 copies. And now, 15 years after the first assay was approved, we're down to single copy detection for HIV. So the amount of effort that's required to do single copy detection of viruses or any targets is dramatic. And then I also show you, in the case of human papillomavirus, the diagnostic primers that first started out with a 450 base per amplicon, went down to 150 base per amplicon, and the currently most sensitive amplicon is only 65 base pair. And that is because in the case of HPV, we need to be able to amplify pieces. What we observed in terms of complete failure, that happens really in the PCR step. So designing the amplicons is more important than anything. And really, you can use the AmpliSeq amplicons and use them in Illumina sequencing and other technologies. That's not the problem. Here we are looking at simple repeats, And so on the top left, you can see if it worked the first time, then simply repeating it will work also. On the next one to the right, we can see that the repeats didn't work, so there's really nothing there. One below in the middle, you can see that we were able to recover something even though the first try failed, and then that's also shown on the bottom right. There is primer bias in diagnostic PCR because simply the primers are designed against the consensus sequence, and if the consensus sequences changes, the primers will fail. This is an example of West Nile virus where we use the same primer pairs and artificially generated oligonucleotides with every possible mutations. And then we ask the question, if we use a reaction where there is a third primer in the middle, which is called the Tachman probe, how many will work? And if we only rely on two outer primers, which is the CT Cyber probe, how many work? And you will see that there are good targets with no changes where both are equivalent. There are targets where the probe fails entirely because the mutations are too big, and there are targets where the sensitivity drops by 10 CTs, which is rather a lot. We also need to be aware that the ends are always missing whenever we sequence viral genome, because these are very difficult to target. In particular, you can't make a primer to capture the 5' end because there is no sequence to align to. And then you can't make a primer to capture the 3' end because either there is no sequence to align to, or it is the poly A tail, which has very different properties than random DNA. So in these cases, we have to invest in 5' and 3' rays, which are very specialized procedures, and they're very slow in that they require about 1 person per week per sample. So the result of that is a problem in GenBank that most people who want to report complete genomes will simply paste the same sequence for the first 42 and the last 61 amino acids and repair that from the consensus. And of course, that is not necessarily true. The ends matter quite a bit. And so here's an example where we identified a deletion in the end of SARS-CoV-2. And in case of SARS-CoV-2, the ends are non-coding, and it's the structure of the RNA that determines function. These were first identified by next-gen sequencing. You can see on the top left a gap. When we artificially generated the sequence to include the gap, you can see that now the reads are spanning the gap. And you can see that by Sanger sequencing, we've confirmed this rather 10 or so 20 nucleotide gap. M-fold gives us the structure, and you can see that in the wild type, in the middle top, the yellow sequence is there. And when the gap is introduced, the mutant no longer has that top left ear. Everything else, however, is correct. So this is very clearly removing a site for something, and that changes the replication of the virus. In the old days, and with DNA viruses, we had to reside to Maxim Gilbert sequencing. And in case of KSHV, that was only done once in 1997. Ever since, all the ends are from that 1997 master sequence. KSHV, this is now more recent work from this year actually, has a lot of good DNA and a lot of DNA that doesn't amplify by PCR. So no amplicon on the way bottom shows you regions of the genome that we could not design primers across. That is, not design primers across that are consistent with the amplification conditions of all the other 300 primers we used. In that case, there's only one way. You have to design specialized primers. You can notice that they're all a different size. They're longer, they're smaller. They have different efficiency if you compare lane number 1 and 2. Lane number 3 also is smeary. So these are suboptimal PCR conditions. They all run at a different temperature. And the only way to get these gaps is to run these individual reactions. Sometimes you have to subclone them. Often we simply put Sanger sequencing primers on the end and then sequence the PCR products. You notice their size goes up to 1.5. And so Sanger sequencing gives you 800 base pairs. And going from both sides, you can then relatively easily go up to 1.5. By comparison, next-gen sequencing gives you only 150 to maybe 400 base pairs. So the length of the Sanger sequencing allows you to span gaps. This is just a summary slide showing you different ways of how these technologies differ: Sanger, Illumina, Ion Torrent, and Nanopore. Those parameters change over time as there's constant improvement. But what we found, and I think what's important, is then that if there is a gap on one technology, Ion Torrent, that same gap would occur in another next-gen sequencing technology. And that's because the next-gen polymerase is designed to work in one fixed condition for all the amplicons, whereas with Sanger sequencing, we have a variety of tricks that we can adjust for every single target. And that is the difficult part. Now, I'm going to go a little bit more quickly to make it within 30 minutes. The question then becomes, how good is our SNV calling, right? And that is the question that most recently came up again when we're trying to define the origin of SARS-CoV-2, which is done by looking at comparative genomics. So this is a number of samples from patients at UNC. There are about, close to 100 samples. The top left, panel A, shows the time since diagnosis. Panel B is more interesting because it compares in the magenta samples that we did generate good sequence, and then in the blue samples where we did not generate sequence. And you can see the shift there that you need a lower CT to generate good sequence. Panel C shows the same again, looking at the magenta versus the blue dots, and we can see the linear increase as the CT is below 28 or even below 20. And that there are very few failures. There are still some failures, and there are still some successes where we get the whole genome out of something that has virtually no nucleic acid, and that's just random variation. And then on the right, you see the graph that you quite often see, which is the percent of genome coverage on the y-axis as opposed to the fold coverage on the x-axis. And clearly you want to have 100% coverage at 1000x. But as you can see, there is a spectrum, and that most of the good sequences will give 100% coverage at 50x, and then it drops off. Clearly something that gives one-fold coverage of only the half genome is not good enough to assemble a genome. Now I want to look a little closer at the SNP abundance in our samples. And here what we're looking is how many SNPs are in each sample. So the top panel A shows that most samples of SARS-CoV-2 have on the order of 12 SNPs. That's the kind of variation we see in the population. On the panel B, you can see if you look at all the reads from a given sample, most SNPs are present at about 100%. So they're fixed in the population. There isn't a lot of subdominant SNPs in SARS-CoV-2, and that is a difference as compared to HIV or hepatitis C virus. The SNPs are distributed sort of randomly across the gene, in that ORF1AB is the largest gene, and then they're ordered S, of course, is spike, so that's under selection. And then we can calculate the distance between any two genome, and that is simply the number of SNPs by which any two genomes differ, and it's a little bit less than 10. So if you look at virus sequences, if we want, and I'm starting on the bottom, a high confidence SNV map, we need twofold minimum coverage, because even for completely covered genomes, individual SNPs, because of the variation, might have high confidence, and we can use the confidence of the neighboring nucleotides to assign quality scores. And so even in a twofold on average covered genome, we are able to identify some SNPs at over 200-fold quality. Complete viral genome, in my view, is 20x minimum coverage. That's the same as for the human genome. And if you wanted to detect minor variants or quasi-species, you would need upwards of 2000x coverage. Here's a direct comparison where on the left, we only look at complete genomes. These are present in GIS8. That's sort of the virology approach, and we can cluster them. And then we took the same data and we only clustered them based on high confidence SNPs by Hamming distance. And now we're able to allocate over 67 samples to a tree, whereas before it was 48. And that's essentially what we do in oncology, in that we're not interested in sequencing everyone's genome. We want to know whether there is a RAS mutation or not. And as long as the sequence coverage across RAS is very high, we can use that information. We don't need anything to the left or to the right of that gene. Finally, the latest data from last week, it shows the evolution of SARS-CoV-2 in the US, or more precisely in North Carolina. And we seem to be having with Omicron a zoo of sublineages, in that the virus is persisting in the population, it's replicating in the population, it's spinning out variants, but none of them are as dramatically different from the original Omicron as Omicron was from Delta. And I think that is a good sign, because you could interpret that the virus has sort of found an equilibrium with the human population, where the transmissibility is high, but there aren't too many surprises in our future. Of course, that's pure speculation, and that is debated by others. Finally, I'd like to acknowledge all the people who did the work. That is my lab on the left. In 2020, when we started, there was no vaccination. There were no drugs. We just had to mask. And then after everyone became vaccinated in 2021, and since, we've been able to enjoy each other's company. Of course, in the lab, we still wear masks. Within confined species and rooms, we still wear masks. And the wearing of masks in a diagnostic lab is actually very important, because the contamination is both ways. It's that we don't want to get infected by the samples, but conversely, if a person is shedding asymptomatically, we don't want that viral variant reported in our clinical samples. So it goes both ways, and the masks protect both the wearer and the environment. And with that, I'm happy to answer any question. Thank you very much for your attention.